The question was how many ATP molecules are generated from one molecule of NADH in the electron transport chain. So NADH actually is produced through several metabolic pathways like your Krebs cycle, glycolysis, beta oxidation. So you have a lot of NADH, FADH2 being produced through these pathways. Now, the question is, in former textbooks, it was always written that three NADH molecules are produced. Three ATP molecules are produced from one molecule of NADH and two from one molecule of FADH2. But recent researchers have shown that they have measured actually how much. They have shown that there is some loss in shuttling your NADH from the cytoplasm into the mitochondria, or from the mitochondria. There is a bit of a loss of energy takes place. So modern textbooks after 2000, all the books after 2000, they all suggest that it is 2.5 ATP, not 3. Okay. So even if you do your calculation as per 3 ATP, they will not cancel. They will give you marks for writing that answer, provided you use that uniformly throughout. If you are taking 3 ATPs, three ATPs for one NADH molecule, then the whole calculation of how many ATP molecules are formed, that should go as per 3 ATP. You can't mix both of them. Then you have to talk that there are 36 ATPs formed. You cannot say that there are 30 ATP molecules formed. So from one molecule of glucose oxidation. So you have to keep it in mind. But I would say that for all convenience, follow the modern nomenclature of 2.5 ATP for NADH and 1.5 ATP for FADH2. Okay?
হ্যাঁ স্যার, তাহলে স্যার একটা টিসিএস সাইকেলে তাহলে তো ওই 10.5 মতো হচ্ছে না,
10,
যদি 2
10.
পয়েন্ট।
না, actually 9 হয়।
আচ্ছা, কিন্তু স্যার যদি 3
একটা
ধরি
হয়
তাহলে
ওইটা পাইরুভেট ডিহাইড্রোজেনেস কমপ্লেক্স। একটা ক্রেব সাইকেলে 10 হয়।
হ্যাঁ স্যার, যদি 3 ধরি তাহলে তো 12 হয়, যদি 3 ধরি।
3 ধরলে 12 হয়, তো 12 টা তুমি নেবে না, তুমি 10 ধরবে।
আচ্ছা আচ্ছা ঠিক
পিডিএইচ
আছে।
পাইরুভেট ডিহাইড্রোজেনেস স্টেপটাকে বাদ দিলে।
আচ্ছা 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 আচ্ছা, ঠিক আছে ঠিক আছে।
ঠিক আছে?
হ্যাঁ।
So basically from one glucose you can expect there are since two molecules of pyruvate, from one pyruvate you get 10 ATP, and so one glucose has got two pyruvate, so you can expect 20 ATPs over there. And pyruvate dehydrogenase you can expect, there is, since there is two NADH, because of two pyruvates are there, so you can expect 5 ATPs over there. So you can expect with pyruvate dehydrogenase 25, and two NADH formed in during glycolysis, you can expect another 5, so it becomes 30. Okay,
ইয়েস স্যার।
so this is actually the calculation takes place. And you can another, there are two ATPs also, one FADH2 is also formed over there, so two FADH2 is also there, so that is another 3 ATPs. So you can go through your calculation. You will see that any standard textbook, I would suggest go through the calculation in the modern Leninger, Delson, Cox. You will find the whole complicated calculations over there. It's nothing very difficult. Okay, let's start today's work. So I will talk about beta oxidation. পেনটা খুঁজে পাচ্ছি না। Let me, I found the pen. Okay, so let me share my screen. Could any of you tell me that if it's visible or not?
Yes,
The
sir.
pink screen is visible
Yes,
or
sir.
not?
Yes,
Yes,
I
sir.
swear.
It is visible. Okay, very good. So then that means I can start my work. So today we will talk about beta oxidation of fatty acids. So first, before we go, let us look at the structure of a fatty acid, specifically a saturated fatty acid. A saturated fatty acid has got no double bonds. A double bond is an unsaturation. So if we look at a saturated fatty acid, one of the famous ones are which comes in the exam is the palmitic acid. Please interrupt me if you are unable to understand something or not. This is the palmitic acid. This palmitic acid has got 16 carbon. Somebody wants to ask something. Somebody says, Nisab has entered, I think, possibly. Okay, 16 carbon is present in palmitic acid. So let us draw the structure of a palmitic acid. COOH. Essentially, this is COOH. But I am writing CO. This is the first carbon over here. Then I go to, let's draw this structure. One first carbon. Now the second carbon. I am not drawing the carbon structure over here. 1, 2. This is 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So essentially, there are 16 carbons. The first carbon is here, first carbon, second carbon, third, fourth. This is very easy. Draw the structure like this, telling loudly in your mouth that this is it. 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So let me draw this part in a bit greater details over here. This part essentially means these two carbons over here. So this is carbon number 2, carbon number 3, and this is CH2. I am not drawing the hydrogen. I am just drawing the skeletal structure. 2 and 3. So this makes the actually it is like it's present like this. Now if we look at this part, the last carbon, the 16th carbon, which is just a dot, that essentially is instead of CH2, that is CH3. In the end, it cannot be empty, so there is an hydrogen over there. So this is the 16th carbon over here. So, are you people clear about the structure over here so far?
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay. Now, another thing is this carbon number 2. In former times, it was also called the alpha carbon. And carbon number 3 was also called the beta carbon. And in the process of the beta oxidation, it is oxidation of fatty acid. At this point, there is the breakage and removal, basically, and oxidation takes place. Place in the beta position. Hence, the process is called beta oxidation. So here there is no double bond, and palmitic acid, this is an even chained fatty acid. Okay, since 16 carbon, 16, it is an even number. It is divided by 2. It is divisible by 2. Clear about the structure of a saturated fatty acid? You people can draw this structure?
Yes, sir.
Yes,
Yes,
sir.
sir.
It is very easy.
Yes,
Just
sir.
draw this skeleton. That's all. Nothing else. You don't have to bother about this complicated structure here. You don't have to bother about the whole complicated structure. Just draw it like this, and you will never forget. Okay. Now let me start with beta oxidation. Okay. Now we will talk about the process of beta oxidation. So, what is the function of beta oxidation? The function is to break fatty acids to acetyl coenzyme A, and this acetyl coenzyme A, as you know, it enters into the Krebs cycle, and it will provide fuel for Krebs cycle or TCA cycle. So, from the beta carbon, this 2 carbon will come out. So essentially, you will have 2 carbon acetyl CoA coming out, released, and the fatty acid chain will reduce by 2 carbon. So the 16 carbon fatty acid will become 14 carbon. Again, another step of oxidation, here say beta oxidation will take place. Again, beta oxidation, it will become 12 carbon. Like this, so on and on and on and on and on. Finally, you'll have only 2 carbon acetyl CoA produced. So, total from 16 carbon, you can expect, that is 8 acetyl CoA released. And that means immediately you can account for 80 ATP molecules. Were you able to understand this? Because each acetyl coenzyme A in the Krebs cycle releases 10 ATP molecules. So if there are 8 acetyl coenzyme A, you have 80 ATP. Understand? Were you able to understand this?
Yes, sir.
Yes,
Okay.
sir. Yes, sir. Yes, sir.
Okay, okay. Very good. Now this beta oxidation, it takes place in the mitochondria of all eukaryotes. Here there is another cycle known as carnitine cycle. Some people call it the carnitine shuttle. The function is to transfer fatty acids from cytoplasm to mitochondria. Its function for beta oxidation. So question comes on the carnitine cycle. Next, what we are going to talk about is what are the beta oxidation connections. Where does the product of beta oxidation lead to? Right. So in beta oxidation, the first step is fatty acid has to get converted into another acyl compound known as fatty acyl CoA. This is the first conversion. So what comes in the inside the beta oxidation? In is Fatty acid as such will not be oxidized. It has to be converted into fatty acyl CoA. What goes inside the beta oxidation? Fatty acyl CoA. And what goes out? What you get out is acetyl CoA. Also, what you get here is NADH it goes. Okay. Now, what are the regulatory steps? First is the primary signal. What induces the primary signal which induces beta oxidation? Primary signal. First signal that now beta oxidation take place. Insulin hormone. Turns off. What does insulin do? Function of insulin is to absorb glucose from the blood. So glucose content is very high in the blood, and so this turns off. If insulin turns off, then the opposite of insulin is glucagon, the other hormone. It turns on. Who else turns on? Where adrenaline. Adrenaline is a flight and fright hormone. So immediately say for example, you are being chased by a bull and you have to run. You immediately need lot of glucose and lot of energy. So immediately what happens? Adrenaline, it needs lots of energy. So when there is adrenaline, immediately beta oxidation is turned on so that more energy is released. Function of beta oxidation, as you know, it is energy. Turns on. Then phosphorylation. If hormones are phosphorylated, beta oxidation, this it will also turn on. These were the primary signals. Remember them. Now we talk about the secondary signals. Apart from this, who are the secondary signals? Malonyl CoA. Basically, you have 3 carbon. Acetyl is 3 carbon, is 2 carbon. Malonyl is 3 carbon. So what it does, it inhibits carnitine acyl transfer. It stops the carnitine cycle. Now let us go to the steps. You people, is it clear with you so far?
Yes, sir.
Yes,
Okay.
sir.
Yes, sir.
Okay, okay. Thank you. Thank you for telling me. Now let us go to the steps of fatty acid beta oxidation. The steps of fatty acid beta oxidation. So let us talk about palmitic acid only. Even chain fatty acid 16 carbon saturated. The simplest case we will be talking about. So let's draw the fatty acid first. This is the first carbon. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So we have drawn a C16, 16 carbon fatty acid, palmitic acid. Now, the first step which takes place is the conversion of this into fatty acyl CoA. An enzyme is present which works. This is known as, step is known as CoA. Fatty acid is going to be converted into fatty acyl CoA. First step is CoASH activation. This is the step which comes in. So always you'll have a CoASH coming in. The CoASH, it comes in. It enters. CoASH enters. Another thing which happens in this process is one molecule of ATP is hydrolyzed into one molecule of AMP plus PPI. Normally, we know that ATP hydrolyzes to ADP plus PI. ADP, but when ATP is hydrolyzed into monophosphate, that means two phosphates are coming out. This is counted essentially as 2 ATP, because when ATP breaks down into ATP, first breakdown you'll have ADP, one phosphate goes out. Then another breakdown, it becomes AMP. So essentially one energy is released here, another energy of ATP is released over here, another phosphate of energy is released here. So this is essentially counted as 2 ATP. So the amount of energy which is required is equivalent to two ATP molecules. Molecules equivalent. Clear? Yes, na bolo.
Yes, sir.
Yes, sir. Yes, sir.
Okay, okay.
Yes, sir.
Okay, great. Now, we go to the next step. So the enzyme which is needed in this step is, I'm writing the enzyme in magenta. It is a long chain fatty acyl CoA ligase. This is a long chain fatty acyl CoA ligase. This is the enzyme which is converted. Now, what does it do? It converts this into SCoA, carbon. Instead of this, it becomes over here and this double bond remains. This is fatty acyl. Then again, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. This is C16 over here. This is a fatty acyl This is a fatty acyl CoA. It is no longer a fatty acid. It is a derivative of a fatty acid. Clear?
Yes, sir.
Now we are going to the next step. What happens? This is known as desaturation. This step. Here, one molecule of FAD becomes takes two hydrogen. Takes two hydrogen. One, if it takes two hydrogen, immediately double bond forms. So let's see it over here briefly. I'm drawing the structure. If the structure was something like this, if you steal two hydrogen from here, it becomes like this. So the saturation goes away and this region becomes unsaturated. I will draw this part in yellow over here. I'll keep it in. Therefore, this process step is known as desaturation. The desaturation step takes place and you have FADH2 formed. Anywhere there is NAD becomes NADH2, FAD becomes FADH2, there always is a dehydrogenase. Because two hydrogen has been taken away and that comes over here. What fatty acid, what dehydrogenase is this? It is acyl CoA dehydrogenase. Acyl CoA dehydrogenase. So this is a fatty acyl CoA. This becomes acyl CoA dehydrogenase, this enzyme. What does it do? It will introduce an unsaturation. Where will it do? Let us draw this again, the structure. It's like this. Don't forget, just keep on drawing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. It's drawn, but the unsaturation, a double bond is introduced. This two hydrogen from between this carbon and this carbon, 2H stolen by FAD. So when it goes, there are double bond forms. So an unsaturation results. And this type of a compound, it is known as enoyl CoA. Because of this double bond, this sort of a compound is known as enoyl CoA. Clear? This step is clear?
Yes, sir.
Okay.
Yes, sir.
Now in the next step, it is known as hydration. Here a molecule of water enters. Water is one H and one OH. This enters. A hydration reaction takes place. Enzyme is, since it is an enoyl CoA, hydratase. This is the enzyme. Now, what happens is, let's draw this again. Structure SCOA. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So the double bond was here. This double bond is removed. Here an H comes and here an OH comes. This is the alpha carbon and this is the beta carbon. Or you can call this as carbon number 2 and carbon number 3. This becomes, the double bond disappears and this particular hydration takes place. This is a hydration reaction, and H comes in one side and OH disappears. So essentially, if I draw the structure in a normal way, you have a double bond C formed over here. If it is like this becomes CH So from water, this has come from water. Say one comes from here and one comes from here. Okay, so this is essentially what hydration has taken place. আমি পেটি এটাতে দিয়ে দেবো ফোন পেতে হ্যাঁ হ্যাঁ So this particular reaction takes place. So clear so far?
Yes, sir.
Okay.
Yes, sir.
Okay, next step after hydration, now comes the, once this OH is over, this forms a target for break. OH is a target for oxidation. This forms a target for oxidation. Now here the final oxidation step takes place. Enzyme is 3-hydroxy acyl CoA dehydrogenase. Here one NAD plus gets converted into NADH plus H plus. Now I have forgotten to mention that this type of a compound is known as 3-hydroxy acyl CoA. Now this 3-hydroxy acyl CoA means in the third carbon there is an hydroxyl group, and in this acyl CoA 3-hydroxy acyl CoA. Therefore it is known as 3-hydroxy acyl CoA. Okay. Now this results in this particular compound. It will steal away this hydrogen and it will steal away this hydrogen again. Result is, let us first draw the structure. This is so easy that I don't need to see. You will never forget the structure if you understand the principle. You don't have to remember it also, if you understand. This is this. Again, you draw this C1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. You have drawn it. Now, in this structure, for example, if you remove this hydrogen again, what will happen? Only the H is removed. This H has been stolen away here, and this another H has been stolen away here. So this particular structure forms, and this OH which was below here, it becomes into this double bond over here. Now this type of one OH is over here. It's a very unstable structure. Already this bond is under pressure to break. This is under pressure to break. This molecular, because this negative charge and this will repel and it will try to pull apart. They will try to push each other apart because of negative charge. So in the next step, what happens is cleavage takes place. COASH is again released. The cleavage will take place here. That means you can expect this unit to come out. This unit comes out. So what you have remaining is this is essentially nothing but this is acetyl CoA. And this O is another COASH will enter inside. Result is this CoA SH, this double bond. This now I am writing just the CoA SH won't stay like that. This I am writing as only the H won't be there. SCOA, this S I am writing ahead. You have the SCOA. This part, this O to this another SCOA is going to attach to this carbon. And this 2 carbon has gone. Now you have 14 left. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So this becomes the new C14. And the COSH comes over here and you have the C14 fatty acyl CoA is formed and this acetyl CoA is generated. This will enter into the TCA cycle. And this will undergo further rounds of beta oxidation. Are you able to see the whole step? Whole step.
Yes, sir.
Now,
Yes, sir.
now what happens? I'm drawing it in blue. This will again go to this step, not the first step. Will go to the second step. Since already fatty acyl CoA has been formed. This is already fatty acyl CoA. Already fatty acyl CoA has been formed. So now the question arises, how many acetyl CoA? (16 carbon). Each step 2 carbon is removed. So total there will be 7 steps. First step 14 carbon, second step 12, third step 10, fourth step 8, fifth step 6, sixth step 4 carbon, seventh step 2 carbon. After that it becomes 0, so it doesn't. So there are 7 times 7 times this cycle will repeat. Now the next question is, how many acetyl coenzyme A? Can anybody reply? Answer.
Sir,
8.
8.
Exactly. So 16 times, if you divide it by 2, you will find 8 acetyl CoA. Next question is then, how many NADH plus H plus? Can anybody reply? Look at the step. One step you get 1 NADH plus H plus. So how many NADH plus H plus?
Sir, 7.
7.
Exactly. 7. Next question is, how many FADH2? How many FADH2?
7.
7.
Exactly 7. 7. So let us see the energy calculation. Energy calculation. So for conversion of fatty acids to fatty acyl CoA, -2 ATP. ATP खर्च है, हो गया. 7 molecules NADH plus H plus. that is 7 into 2.5, that is equal to 10.5 ATP. Then we have 7 molecules of FADH2. 7 into 1.5, that is
सर ऊपर वाला 17.5 होगा नीचे वाला 10.5
हाँ 17.5, ये 10.5 हो गया. Absolutely right. This becomes 17.5. ATP. And 8 molecules of acetyl CoA. This is, 8 into 10, 80 ATP. So if you add the total, 108 minus 2 ATP, which is being used here, 106 ATP. 106 ATP formed. So in the next class, we will be talking about what happens in unsaturated fatty acid and odd chain fatty acid and beta oxidation in peroxisome. Oxidation in peroxisome. And also we will be talking about the carnitine shuttle. ओके, परसों ये करेंगे, ठीक है?
Okay, sir.
ओके सर ओके सर
Okay, I've stopped sharing. So do you people have got any questions?
স্যার ওই স্যার রেগুলেশনে ওই ফসফোরাইলেশনেরটা স্যার একটু বলবেন স্যার ওই
Enzymes get
bit-
phosphorylation and dephosphorylation. This is a process by which enzymes get activated and inactivated. So if the enzymes are enzymes of the beta oxidation, like you have seen several of these enzymes. Oh, I forgot to mention. Let me show you. I have not written. What is the enzyme which this cleavage, this step of cleavage which takes place here, right? This cleavage enzyme is beta
স্যার
thioylase.
স্ক্রিন ইজ নট ভিজিবল
স্ক্রিনটা ভিজিবল নয়? আচ্ছা, শেয়ার না করলে ভিজিবল হবে কি করে? নাও ইট শুড বি ভিজিবল। এই যে this cleavage, COASH, this is beta thiolase, this enzyme. Okay. I forgot to mention about this enzyme. So enzymes are activated and inactivated by phosphorylation. So phosphorylation activates it. That was the question. That was what it is. Any other questions? So then we meet day after tomorrow then?
Okay, sir.
Okay, sir.
Okay.